Objective: to study about human reproduction, oogenesis, Part 1. The process of formation of the female gametes, that is ova, is known as oogenesis. Oogenesis is markedly different from spermatogenesis. Oogenesis is initiated during the embryonic stage of the female. It consists of 3 stages. They are multiplication phase, growth phase, and maturation phase. Multiplication phase. During embryonic development, the primordial germ cells present in the germinal epithelium lining the ovary produce oogonia by repeated mitotic divisions. A couple of million oogonia are formed within each fetal ovary. By the time the female fetus is 20 to 25 weeks old, all the oogonia are already formed. The oogonia do not undergo further multiplication. Growth phase. During the growth phase, the size of the oogonia increases many times. Maturation phase. It is the longest phase in oogenesis. The oogonia enter into the prophase stage of meiosis I. However, further development is suspended at the diplotene stage. In this stage, they are known as the primary oocytes. Each primary oocyte is surrounded by a layer of flattened epithelial cells. This structure is known as the primordial follicle. The flattened cells surrounding the primordial follicle become cuboidal. These cells then proliferate to form 2 to 3 layers of cells. These cells are known as the granulosa cells, and the layer is known as membrana granulosa. This stage, with the primary oocyte surrounded by the granulosa cells, is known as the primary follicle. A large number of the primary follicles degenerate between birth and puberty. This is called follicular atresia. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. With the formation of the theca, the follicle is referred to as the secondary follicle. A fluid-filled cavity called the antrum or follicular cavity develops between the granulosa cells. The fluid present in the antrum is called follicular fluid or liquor folliculi. It contains nutrients and estrogens secreted by the granulosa cells. The oocyte now lies in an eccentric position within the follicle. The follicular cavity increases in size. As a result, the granulosa cells are differentiated into 2 layers. The thin layer surrounding the primary oocyte is called cumulus oophorus or discus proligerus or germ hill. The outer layer remains as the membrana granulosa. The follicle with antrum is known as the tertiary follicle. The primary oocyte within the tertiary follicle completes meiosis I. This is an unequal division. It results in the formation of 2 haploid cells. The larger cell is the secondary oocyte, and the smaller one is the first polar body. The secondary oocyte is large as it retains the bulk of the nutrient-rich cytoplasm of the primary oocyte. The secondary oocyte enters into meiosis II, but the development is suspended at metaphase II. A homogeneous layer called zona pellucida appears between the secondary oocyte and the granulosa cells. Zona pellucida is a non-cellular glycoprotein layer secreted by the secondary oocyte. The innermost layer of granulosa cells is firmly attached to the zona pellucida. This layer of cells is called corona radiata. The theca around the follicle differentiates into theca externa and theca interna. The theca interna is made up of stromal cells. The theca externa is made up of fibrous stromal tissue. The follicular cells, along with the cells of theca interna, secrete estrogens. This is due to the stimulatory effect of the follicle-stimulating hormone FSH. The tertiary follicle further grows in size to form the mature follicle or Graafian follicle. As the follicle increases in size, it forms a bulge on the surface of the ovary. The Graafian follicle ruptures to release the secondary oocyte or ovum from the ovary. This process is called ovulation.